everyone. Welcome and greetings. This is an exciting opportunity for us with the KIBM Symposium this morning and the Institute for Neural Computation retreat in the afternoon. KIBM has been partnering with INC for a number of years now. We enjoy these meetings tremendously. Of course, this is the first time we've been doing it as a Zoom, but many of us have been doing a lot of Zooming. So I think that this should go very well. And Megan Ensminger is helping us out with a lot of the logistics. So I think she and others really got us up and running here. This is the 15th year of awarding innovative research grants. It's quite a milestone. I want to offer congratulations to this year's awardees. There were 10 of them. You can find them on the KIBM website. And tough to make these awards. There were 40 proposals. We'd have been delighted to fund another 10 or more, but there's only so much money to go around. So we're of course working hard to try to increase the endowment, but these were great proposals and we're very happy about that. I want to take a quick moment here to recognize the incoming co-directors of KIBM. Rusty and I will be stepping down in a few days, and Yishi Jin from UCSD and Ed Calloway from SOC will be stepping in, and we're very excited about that. This is going to be terrific. In addition, I want to recognize the two associate directors, Ralph Greenspan continuing from years past, and importantly, Gabe Silva from the Jacob School will be joining us. And we think this is wonderful because we've seen a growing connection to the Jacob School and engineering, and this really strengthens that relationship. So this morning in the symposium, the KIBM Symposium, we'll hear from 8 of the last year's awardees. Exciting science. Again, as you know, we're trying to bridge brain and mind here, reaching out to bring these two camps that had in the past been somewhat separate, trying to integrate them. And I think this has been very rewarding. The format, as you know, involves a 10-minute talk and then 5 minutes for Q&A. And please type your questions into the chat. There's a little icon at the bottom. If you're not familiar with this, you just click on that and then a screen appears on the right, type it in. And then I'll be looking at that, and we'll hope that the speakers will leave some time for us to do the questions afterward. So I think with those formalities out of the way, let me turn to our next speaker, Amy Pribadi from Biological Sciences and the Salk Institute. And Amy's going to get in here and share her screen when Gerald gets off there. Thanks a lot, Gerald. And she's going to talk about a new way of monitoring temporal information flow in C. elegans. A beautiful story here. So Amy, please take it away.
Hi, yes. Thanks everyone for coming to my talk this morning. I'm a graduate student in the Chalasani lab at the Salk Institute. And today I'm going to talk to you about my project using CaMPARI in C. elegans and how I hope to use it to monitor temporal information flow during complex behavioral experiments. So the main model organism of choice in the Chalasani lab is C. elegans. C. elegans is a convenient model for the neuroscientist because all of its 302 neurons and their connections have been mapped, and most are genetically accessible through cell-specific promoters. It's also clear, meaning that fluorescence can be imaged in a live animal. This is a C. elegans adult with all its neurons expressing a fluorescent protein. There's a dense cluster of neurons near the head of the worm, which we loosely refer to as its brain, and this contains most of the worm's sensory neurons and interneurons. So one of my overall goals is to identify shared neural circuits in similar complex behaviors. C. elegans navigates the world mostly through its sense of smell and are able to remember positive and negative associations to specific odors. They then can modify their behavior depending on past experience, either moving toward or away from that odor. For example, C. elegans can negatively associate odor with starvation or with toxicity of food. I'm interested in how this behavioral plasticity can arise. My next and related goal is to record and compare neural activity over multiple periods of time during different epochs of these behaviors. For example, I could compare a worm just starting to learn to a worm at the end of its training period to one that has already forgotten its training. To study neural activity in complex behaviors such as these, I thought that CaMPARI would be a useful tool. CaMPARI has been developed in multiple models, including flies, zebrafish, and mice, but not C. elegans. I will first need to validate that CaMPARI works as expected in C. elegans by testing its light conversion specificity and its activity dependence. Finally, to compare across multiple worms, I need a way to identify single neurons. After the validation of this technique, I can then apply it to behavior in an often used assay that I will discuss later. To study neural circuits, most researchers record videos of genetically encoded calcium indicators such as GCAMP. A single worm would be immobilized in a microfluidics chip such as this that allows control of stimulus delivery directly to the tip of the worm's nose. This technique has been used in many studies and is a powerful way to study neural responses to precise stimuli. However, the types of experiments that this can be used for are limited because the worm is paralyzed and cannot perform any behavior. In addition, this technique is not amenable to long-term studies as the worm cannot eat and the excitation laser will bleach the fluorophore that you're measuring. CaMPARI is a tool developed by Eric Schreider's team at Genelia. It starts out fluorescing green, but switches to red in the presence of high calcium and near UV light. In order to record total activity over a set period of time, researchers need only to shine a 4 or 5 nanometer light on a population of neurons for that period of time. After photoconversion, the neurons can be imaged in a single still image. This sacrifices dynamic data over time, but allows for more detailed imaging in the later step, since imaging does not have to be done quickly for the video. The in vivo activity-dependent photoconversion of CaMPARI was validated in intact behaving zebrafish larvae by comparing the brains of anesthetized, freely swimming, and seizing zebrafish. They showed that anesthetized zebrafish barely had any red photoconversion, while inducing a seizure promoted widespread red photoconversion. Freely swimming zebrafish were somewhere in between. To identify neurons, I'll use a tool developed in the Hobart lab called NeuroPal. This is a worm expressing many different neural reporters, expressing one of 4 different distinguishable fluorophores in the nucleus. Each fluorophore gets assigned a pseudocolor, and the resulting overlay creates a worm with neurons with stereotyped colors. This worm also has a pan-neuronal nuclear red marker as a landmark. So first, I created a plasmid carrying a C. elegans codon-optimized CaMPARI under control of a pan-neuronal promoter. I injected this plasmid into the NeuroPal strain and created a worm with nuclear localized NeuroPal reporters plus my cytoplasmically expressed CaMPARI. And this is showing the worm's brain, as I talked about earlier. I tested to see if this codon-optimized CaMPARI would still only specifically convert under 4 or 5 nanometer light. The worm on the left has had no photoconversion light exposure, while the worm on the right has been exposed for 30 seconds. The red is undetectable in the worm without the photoconversion light, confirming that this CaMPARI does only convert when exposed to 4 or 5 nanometer light. I then wanted to test for the overall activity dependence, similar to how the Schreider lab validated CaMPARI in intact zebrafish larvae. For the high activity condition, I photoconverted freely, moving worms on food. For the low activity condition, I put worms on a cold agar plate without food. This cold anesthetizes the worms temporarily. I expected to see overall more red CaMPARI in the worms on food and less in the worms on cold. Here are some results I was able to obtain before the imaging core at Salk shut down due to COVID-19. As a reminder, CaMPARI has been injected into a NeuroPal background, which includes a pan-neuronal nuclear marker that fluoresces similarly to the red CaMPARI. However, CaMPARI is expressed cytoplasmically only. I measured total normalized red fluorescence by dividing the red signal by the green signal. And I found that cold anesthetized worms had lower normalized red than those on food, confirming that CaMPARI conversion is activity dependent in C. elegans. Looking at the raw green and red values, the green values are variable in both conditions as a result of CaMPARI, the CaMPARI transgene being an unstable extrachromosomal array. The red, however, closely clusters in the cold condition, since this is mostly due to the red integrated neuropal reporter. The red in the food condition, however, is more variable since the red photoconverted CaMPARI got added to the neuropal signal. It's been shown that C. elegans can undergo aversive olfactory conditioning based on the presence or absence of food. As shown in this diagram, C. elegans is normally grown in a lawn of bacterial food, represented by this brown. A population of C. elegans can be exposed to a single, mildly attractive odor, such as to butanone or benzaldehyde, represented by blue, either in the presence or absence of food. After some time, the population is then tested for their preference to this blue odor in a chemotaxis assay by transferring them to the center of an odor gradient. Worms trained with the odor in the presence of food will find the odor more attractive than normal, while worms trained in the absence of food will find the odor more repellent than usual. The robustness of this behavior shows dependence on training time. Worms trained for 30 minutes show weak learning, while training for 90 minutes shows strong learning for 2 hours. Using a spaced training paradigm, long-lasting memory can be made up to 16 hours. I conducted this experiment with an hour-long exposure to benzaldehyde to make sure that I could replicate it and that neuropal and CaMPARI do not affect behavior. I was able to replicate the behavior in wild-type worms, but while the CaMPARI worms showed a decrease in attraction in the trained worms, this difference was not significant. And this was probably due to large variability of the results because I had to eliminate several trials where not enough worms were in the assay due to low brood size of neuropal and the incomplete transmission of the CaMPARI transgene. So to get more consistent results in the future, I would redo this experiment with CaMPARI integrated into the genome. In the future, I'd like to work on fully utilizing neuropal for cell ID. There were some unforeseen problems with overlapping channels in the CaMPARI and neuropal, which can be helped by tweaking expression levels. In addition, I've created plasmids containing CaMPARI2, which is a newer and more specific CaMPARI, and a reversible CaMPARI. I'll also need to optimize photoconversion for maximum specificity, depending on how long the photoconversion light is on. Finally, an interesting application of this technique could be to conduct activity-dependent RNA sequencing by sorting and isolating the active red neurons. A protocol for neuron-specific sequencing based on fluorescence was already developed by the Murphy lab. This would be an interesting way to identify candidate genes that might otherwise be masked by sequencing irrelevant tissue. Finally, I'd like to thank the Kavli Institute for funding this project and my PI, Shrek. I'd also like to thank Eric Schreider, who provided me with CaMPARI reagents and advice, and Javier Hao, who just defended, and he helped me with neuropal imaging. And finally, I'd like to thank the Hobart Lab for giving me neuropal and for the development of all its online tools for analysis. And with that, I will take any questions.
Thank you, Amy. That's just marvelous. It's beautiful, that technique, and very powerful. We'll have some time for questions. Thank you for being careful about that. So let me start off by asking how, so you have 5 distinct recognizable fluorophores. Does that mean you look at 5 neurons at a time, or can you do that to look at really as many neurons as you would like to look at?
Yes, so with neuropal, you're actually looking at all of the neurons in the C. elegans. The fluorophores are expressed in different levels in a stereotyped way in each neuron. So the Hobart lab has sort of figured out how to identify each neuron based on the level of each protein.
Great. Good.
Yishi Jin has a question. Jin,
Yeah.
unmute yourself.
Yeah, I have a question. Amy, great talk, nice work. Just a couple clarifications. You're driving CaMPARI with which promoter?
CaMPARI is under RGF1 promoter.
And then when you did the cold assay and
yes.
shell defect, do you see, is the goal to showing that the worms don't move, so you're seeing changes in the locomotion circuit?
Yeah, so I was having some trouble finding an anesthetic that specifically eliminates like sensory neurons. So I figured cold would be a good way to suppress overall neural activity. So this was really just a general, like, does CaMPARI change at all when activities change? So I didn't really look at specific neurons types in that experiment.
And if I can follow up one more,
Yeah.
is Oliver Herbert also had one that has all GCAMPs that's expressing every cell.
Yes.
And under that cold condition, does it match the calcium levels?
That's a good question. I haven't tested that actually. We do have that strain and other people in the lab are working with it, but I haven't looked at it.
Great, thanks.
Thank you.
Amy, let me quick follow up on one of Jin's questions. So it looked as though, again, as you know, I'm not an elegans guy, but an aficionado. And it looked as though the transfection for the codon optimized CaMPARI was pretty efficient. Are you getting, all the neurons or 90% of the neurons in this imaging?
Yeah, so I can't really speak to the percent of neurons that are expressing, but the nature of these transgenes is they're actually a mosaic because they are not integrated into the genome. So I probably won't have 100% of the neurons expressing CaMPARI, which is another reason to integrate the gene.
Yes, yes. And finally, could you say a bit more about how you're going to do the future, pursue the future correlation between the activity and the behavior? You led us down that road in the beginning of your presentation, and I suspect we're all eager to know how you think that will unfold.
Right, so I was going to talk about a few other behavioral assays I was interested in, but in the interest of time, I cut them out. My idea would be I would look at different sort of aversive olfactory conditioning paradigms, and then I would photoconvert at sort of similar times for each behavioral assay, and then I'd compare across to see what similarities and differences there are in the total neural activity.
Great. Great, wonderful. Well, look, thanks again, Amy. This was really nice, and appreciate your presentation. It was beautiful. So let me turn to Robert Loughnan from Cognitive Science at UCSD, and he'll speak to us about biologically constrained deep learning methods to predict complex human traits from genetic data. So.
Hello, yeah, thank you, Nicholas, for the introduction. Yeah, as Nicholas said, I'm Robert Loughnan. I'm a PhD candidate in the Cognitive Science Department, and I'll be talking to you about the project that I've done over the last year, specifically building a biologically constrained deep learning model to predict complex human traits from genetic data. Motivation for this work is that many traits are predicted to have high heritability, so that's that they have a large genetic component, yet genetic studies only are able to explain a small fraction of this heritability. This phenomenon, where there is a divergence between the theoretical heritability and what we estimate in genetic studies, has been termed the missing heritability problem. And some argue that it may be due to the fact that genetic studies do not model the interaction between genes, or which is known as epistasis. So if we could predict diseases allowing for known interactions, we may, 1, improve prediction, and 2, enable biological interpretation of models. So to overview how genetic studies are conventionally done, you take a number of individuals, you genotype them, and then here in red are shown variants that are variable in the population. You then might measure a phenotype of interest, say, for example, individuals who have schizophrenia versus ones who do not. And then you'll be learning effect sizes for each of these variants with reference to the phenotype. So here are 4 different effect sizes, and these would be learned without any modeling of interaction with each other. And so this has been a very powerful approach, and it's uncovered a lot of biology of complex traits. However, as I said previously, it does not go, and it often doesn't explain the full theoretical heritability that we expect. I'll refer to this approach as univariate regression. So one way in which we might want to consider modeling the interactions between genes, which the univariate approach does not, is we could use neural networks. And these have been applied in many different areas and shown to be very powerful. However, they are famously difficult to interpret how they are learning their associations. One way in which we could constrain this is to use gene ontologies, which describe how, this is about 20 years worth of work, which describe how genes interact with one another into functional groups. And so thereby we would make sure that the architecture of the network was only connected where there was underlying literature which supported that certain genes grouped with one another interacting into functional groups. Therefore, the input to this network would then be genes, and each of the nodes would no longer be hidden nodes that we would not really understand. Instead, they would be related to directly annotated genetic interactions that are supported by literature. So for example, if we were associating genetics with schizophrenia, we could then, after the model was learned, look at how the model was associating genetics with schizophrenia and see where larger activations were related to the phenotype of interest. And then we could interpret by looking at those nodes. Say, for example, this node could be synaptic pruning if we were looking at something like schizophrenia. So this approach I will refer to as D-cell, as it has been, is based on previous work, which was done in yeast 2 years ago in the IDECA lab here at UCSD. So they built exactly this sort of model where they had a neural network architecture that was constrained by gene ontologies. The inputs for this network were gene deletions that they produced experimentally, and then what they were recording in the yeast was the phenotype of cell growth. And so they trained this model to predict cell growth from these double deletions, which were experimentally done. And what they showed was that this model here shown in blue had very good performance when compared against these other models in right. But critically, they also showed that they could interpret this model. So here are two gene deletions, PMT1 and IRE1, which resulted in lower cell growth of the yeast. So they could predict that. And then when they actually looked to interpret the model, they could see the pathways which were being implicated, and they could specifically implicate unfolded protein response in this pathway. And then this was further validated experimentally. So this was performed in yeast, but this has not yet been applied to humans. And so the goal of this project is to build a D-cell-like framework to predict complex human phenotypes from human genetic data, to train weights to predict those phenotypes, and then if the network is shown to have good prediction, interpret the weights and validate previous known pathways for disease as well as new ones. I ideally want to apply this to psychiatric disorders like schizophrenia and bipolar disorder, but due to larger sample sizes and being well powered, as well as a biology that is reasonably well understood, we're currently profiling this in blood lipids. Blood lipids in their own right are interesting to study. They've been strongly implicated in cardiovascular disease, stroke, diabetes, as well as Alzheimer's disease, all of which are leading causes of mortality in the industrialized world. They only have moderate heritabilities around 50%, and specifically if we're interested with the brain, genes such as ApoE have strong signals in predicting blood lipid levels as well as Alzheimer's risk. The data set that I'll be using is 50,000 individuals from the UK Biobank. The inputs which I'll be using are exome level data. And then the phenotype which I'll be predicting is triglycerides over HDL cholesterol, which has been implicated in insulin resistance as well as cardiovascular disease. So one of the first steps of this project is to represent the features, these input exome data in a meaningful way. For what was previously done in yeast, we had these gene deletions, and so that allowed a one-to-one mapping for the inputs for gene ontologies where the terminals are genes. In human level data, we rather have, for this specific data set, we have exome reads. So this is a single gene where we've sequenced each base, and we have certain locations where there are variants shown here in red. And so to get something akin to a gene deletion, what I've done is computed a combined annotation-dependent depletion score. And this is a previous well-validated method. These are pre-trained models that are used to predict how deleterious a variant is or how harmful it is. So each variant therefore gets a number, and this deleteriousness is trained on other assays that don't have any knowledge of my specific phenotype. And then what we do to get an input to this gene ontology neural network is, as it's expecting genes as its input, we take the max score as the representative score for each gene for each individual, and that becomes the input for them into the network. So to overview the pipeline, we take 50,000 individuals from UK Biobank with exome sequencing data, compute CAD scores for each variant, taking the max for each gene, that's 18,000 genes. We then compute the ratio of triglycerides over HDL for each individual. We correct for age, sex, top 10 principal components of genetics, and then we train a model to predict this ratio in 5-fold cross-validation. And I'll show you the results for firstly univariate regression and then D-cell. And I will show you models where we could include all the total gene set, so all 18,000 genes, or a subset of the genes where these genes have been ranked by the univariate association with the phenotype of interest in the training set. And that's what's shown here. So on the x-axis, you can see on the right-hand side is when we include all 18,000 genes. And then as we move further to the left, we can see models where we have a subset of those genes. And there are the two models here, D-cell and univariate regression. So the first thing to note is that both models achieve reasonable performance. The signal is also sparse, is something to note, with the best performing models being where we include about 200 genes here. So this indicates that the signal is sparse and that we may benefit from regularization techniques. So what I'll show you on the next slide is these two models at the top 200 genes being compared with models where we include all 18,000 genes and have regularization techniques. So here is that those top 200 performing genes, those two models I showed you in the previous slide. And then on the right-hand side is where we have a model where we include all 18,000 genes. For linear regression, there is no regularization. You can see that's quite poorly performing. And then ridge and lasso, where we induce regularization of the models. So linear regularization slightly outperforms D-cell, and so this suggests that D-cell may benefit from regularization, and that's ongoing work. But in terms of jumping ahead, because the real kind of selling point to D-cell is that you can interpret this model. So to get into some of the details of that, if we look here, if you remember that each of the nodes within the network correspond to gene ontology terms, and so what we can compute for each node after the network has learned its association is something known as the RLIP score, which stands for Relative Local Improvement in Predictive Power. We'll compute, how you compute this score is you compare two models with the parent and the children. The first model is where you predict the phenotype from the parents, and the second model is where you predict a phenotype from the children. And then you calculate the ratio between these two. And so the intuition behind this score is that it gives you, for each node, the increase in power you get from modeling the interactions between genes. So the larger that score is, the better that node is at modeling the interactions between genes in terms of increasing predictive performance. A number greater than 1 means that it's doing better than its children. A number lower than 1 means that that node is doing worse than its children. So we can rank each of the terms on the x-axis here according to that score, and then on the y-axis is the score itself. And so you can see that there's a very small set of terms which seem to be benefiting from modeling interaction. And so if we look and zoom in, these are relatively surprising results. They could possibly implicate novel pathways that are related to these blood lipid levels, but these are relatively preliminary results. And so more work needs to be done to investigate whether these are false positives or truly novel pathways. So some concluding remarks. I've shown that constrained deep learning framework can be used to predict phenotypes and understanding biological pathways. Future steps include regularizing the network to improve performance and simulations to profile the model, as well as testing on brain-related phenotypes, as well as ones where we may expect epistasis. So thank you very much for listening. I'd like to thank the CAVLI for their funding, as well as these researchers for their assistance, and I'd like to invite questions as well.
Thanks very much, Robert. That was fascinating, just fascinating. Just as a quick comment, I think it's marvelous to be able to actually try to interpret how the neural network is performing by actually specifying, constraining each layer in the network. That I'm sure this is probably now the state of the art, but I had not been familiar with that. Here's a quick question: how can you say a bit more about the normalization, the regularization process introduced with Ridge and Lasso? They clearly made a big difference there.
Yeah, so this is D-cell here. So this is the model, which is kind of the main part of the project. And then all of these models to the right hand are various linear approaches. They don't model the interactions. So these two on the right hand side are ones where we include an L1 and an L2 penalty for performing that linear regression, but it includes all of the 18,000 genes. And so, yeah, it's just in the training set, we sort of do cross-validation to find the optimal alpha in a relatively conventional sense, and then use that alpha within the test set. But what I'm quite interested in doing is, if we can get D-cell to regularize, because it definitely does seem like a sparse signal, then it would be ideal if we could get this level of performance, or hopefully greater, but still include all 18,000 genes, so that when we're interpreting the model, we can consider all potential pathways that could be implicated in the specific phenotype of interest.
But it was interesting that you got your best performance initially with just the 200 top genes. And then as you added more, now of course without the regularization, the performance went down. Does that argue for, in the future, for trying to find better ways to select key genes to keep them small?
Yeah, it does. It's just the way in which these top 200 genes are selected is that they're selected with their kind of univariate association with the phenotype of interest. And so it's kind of, if I'm specifically searching for genes where I want to find interactions, then it's maybe kind of quite simplistic ways to be searching for it. So I'd ideally like to have a more flexible way in which I can find that small set, because I'm specifically trying to find interactions between genes.
Yes, and maybe a final question in D-cell. What were the layers? They weren't in your slide. They weren't identified. I wondered if were those constrained, or how were they brought in?
Oh, sorry, for this model here, how are they constrained?
For D-cell.
Yeah, so those were taken from Gene Ontology, which is a large database.
Yes, right. Okay.
So that's specifically corresponded to each of those layers. That hierarchy was exactly mapped onto Gene Ontology's database.
Great. So Eric Halgren has maybe a quick last question here. Eric asks, can you give an intuition as to why you would get worse performance from parents than from children? The point being that interaction might be expected to give you a better performance.
Yeah, to be honest, I've been racking my brains a little bit about that as well. I'm not too sure. If anyone has any suggestions, that'd be great. But I guess it's maybe something in a kind of a non-optimal way in which these weights are bringing forward the signal from these lower categories. And there's also going to be some, I can't really go into the details of it here, but there's going to be some loss of information. So this is a lower dimensionality than its children. And so some of that information is going away. And so possibly that it's just doing it in a non-optimal way, those nodes.
Great. Well, hopefully people will get in touch with you and offer you some further suggestions there. But again, let me thank you, Robert. That was great. And we'll turn now to Dhruv Grove. We'll turn now to Nick Gravish from MAE, Mechanical and Aerospace Engineering, up at the Jacobs School. And Nick's going to tell us about three-dimensional tracking of flying insects at high spatiotemporal resolution.
Great. Thank you, everybody. I'm assuming everybody can hear me. I'll just go ahead and get started. So thank you. This is my first time, interacting with folks at the Kavli and KIBM, and I'm really happy with the work that was funded here. The title of the talk, Three-Dimensional Tracking of Flying Insects at High Spatial Temporal Resolution, has been slightly modified in that we've really been working with walking animals so far. So that's just a little caveat. But the overarching motivation for this is that in my lab, we're interested in studying animal movement as well as creating bio-inspired robots that are sort of inspired by the capabilities of animals. And in particular, I'm interested in the challenges of natural environments, the kind of locomotion challenges that realistic natural environments with complex sensory inputs and sort of locomotor challenges present to animals. And previously in my work, we've sort of taken efforts to try and understand flight and walking behaviors in what I call kind of naturalistic environments. So in some experiments, we've looked at the flight of bees who are freely allowed to go out and forage in the natural environment, but have to fly through complex structured arrays of obstacles and try and reconstruct, reconstruct the type of locomotion control inputs and sort of sensory inputs that are required to navigate these kinds of environments. The sort of techniques that we use for this kind of imaging rely on multi-camera, three-dimensional tracking and sort of modest high-speed tracking of these animals performing these movements. And really the ability to observe these animals moving through these sort of naturalistic environments has allowed us to identify interesting novel behaviors associated with, for instance, like right here, navigating around obstacles using sort of lateral side slip type behaviors in flight. But one of the challenges is that if we're interested in understanding those kinds of behaviors, we really want to be able to zoom in and look up close at what the particular sort of head movements are to understand their field, the bee's field of view, understand the wing movements so we can reconstruct kinematics. And that presents a challenge. These sort of wide field observations are confronted with the challenge of relatively low resolution. Focusing on ground-based animals, we can do a little bit of a better job because we can at least know that they're moving in a single plane. And so, more recently here at UC San Diego, we've been doing work with looking at legged locomotion on kind of more, laboratory complex environments. So sort of three-dimensional structure that has some regularity to it. And again, using multi-camera imaging techniques to capture, again, kind of modestly high-speed videos of these movements. And even from these videos, we can do a lot of great things in terms of tracking the animal's kinematics, full pose kinematics using techniques like deep learning. But we're still, there's still something missing in our ability to reconstruct these kinds of movements over large spatial ranges, as well as at the kind of highest temporal ranges that we would like, thousands of hertz of high-speed recording, specifically for flying animals, where we really want to understand the details of wing movements as well as body pose and positioning. So this is where Dhruv comes in, who is thankfully just gave a talk, and I don't really need to go into this much, but Dhruv here at KIBM has developed flyception, a technique for doing really high resolution imaging, specifically of the brain in this instance, using a mirror-based system to drive the focal plane of a high-speed camera to follow around an animal. And with Flyception 2, Dhruv was able to implement sort of Z tracking so that you can now observe not just the animal moving in X, Y, but also moving in Z as well. But again, I think that the tracking within Flyception is that the animal's still moving in a relatively featureless environment. And that's not a downside, that's a sort of necessary feature, I believe, of this kind of, brain imaging. But what we wanted to do as part of this KIBM proposal is to basically smash these two techniques together. Okay, can we use wide field 3D tracking to observe animals moving in structured environments? And can we then use galvo-based tracking to direct a high resolution, high speed camera system towards that animal that again is moving in this kind of like structured three-dimensional environment? And so, wide field tracking has its downsides at relatively low resolution and just modest speed. And galvo-based steering of the focal plane has its downsides in terms of relatively limited arena sizes in sort of current form. So Flyception 3D is what we've been working on. And this talk is largely a tool building talk where we've mashed together redundant multi-camera 3D tracking, high-speed galvo target tracking, and electronic modulated autofocus. And in principle, we're shooting for using 4 or more cameras to 3D track in real time, and I say real time at 100 hertz streaming through a computer and outputting the instantaneous XYZ location of the moving animal. We then steer an overhead galvo to track the animal and focus at the specific Z focus that we need. So this is what the system looks like. This was its current form with 4 machine vision cameras that are doing 100 Hertz XYZ tracking. The XYZ tracking position is then sent to the galvos. The galvos are aligned such that a high-speed camera is viewing the animal, and we use a LED backlit recording arena for sort of simplicity of tracking in its current form. Like I said, we are using these machine vision cameras. We chose 4. We have plans to add on more for as a sort of minimum, well, not minimum, but the minimum that we wanted to work with for doing this tracking. The Galvo system was designed and assembled as part of a UC San Diego Mechanical and Aerospace Engineering senior design team that machined some custom components for this system. And in principle, it sits about 1 meter above the arena with a 120 millimeter camera lens focused through the Galvo. This is just some details of what it looks like. And again, we're using kind of a very boring LED backlit observation arena for our testing so far, with the goal of through the redundant multi-camera tracking, being able to add in structure to this environment and still be able to reconstruct the 3D position of the animal. Some details on the tracking. You know, there's nothing terribly new that we're doing here. It's more just, again, putting together a multitude of different algorithms and techniques that have been independently developed for these systems. We're doing image acquisition via triggered cameras, 4 cameras in principle, that are streaming at 100 Hertz, background subtraction happening at 100 Hertz, and we're detecting the animal at 100 Hertz as well. The points from the animal get fed into a triangulation algorithm that's relatively standard. And out of that, we get the XYZ position of the animal with respect to the arena. Some conversion converts that point to the Galvo coordinate system. And as Drew said, the Galvo takes in 2 inputs, basically 2 angles that steer the mirrors so that they can point to the desired target. And then there's a third signal, a Z signal, which uses the same sort of hacked focusing technique that Flyception 2 uses to focus the lens. So we can look at a demo of this. So we're going to track just an LED that's moving in space. The Galvo is tracking this LED based on the tracking within the 4 machine vision cameras. And this is just a proof of principle that we can image over a relatively wide area. This LED is just moving in the Z plane, sorry, at the Z equals 0 plane. And you can see an example of this moving at a sort of higher level here. So again, just proof of principle that we can track a sort of very apparent object in LED that is clearly able to be seen by these 4 cameras. Let me show you an example with an actual animal here. So here's a cockroach, Blabberus discoidalis. We've got a little tag on the back of it for identity tracking. Again, this animal is moving in a featureless environment. It's just a sloped plane so that we could test Z focus as well as the XY focus. What you can see on the bottom right is the real-time tracking from the Galvo, which sort of tracks relatively well. There's improvements to be made for sure, and I'll talk about that in a bit. And then on the left, you can see details from the high-speed camera. This is recorded at 2,000 frames per second and replayed 100 times slower. And so, you know, I'll talk about some of the caveats with this, but just to give a kind of overall picture of what we've been working on, and then I'll step back and talk about some of the future work. The overall idea is to design a wide field arena for multi-camera animal tracking using kind of real-time techniques for XYZ tracking, sending that to the Galvo so that we can get high resolution, high temporal recordings of animal movement, and using lens focus communication that is something that is relatively new. The next steps are obviously flight, as the title implied. We've been sort of working at a snail's pace with the COVID situation, as everybody else can imagine, and in particular, experimental work has been greatly slowed down. But I want to just step back and also focus a little bit on the details that we're showing here. You can see that we've got a bit to do in terms of improving the tracking in the context of an animal that does not have fiducial markers like the fiducials that Drew is able to place on Drosophila. There's no reason we couldn't put fiducials on these animals. However, in flight situations, the change in pose of the animal might present significant challenges for using these kind of retroreflected markers. So we need to improve feature detection, feature filtering within the cameras, individual camera streams, triangulation, filtering of the XYZ, lots and lots of filtering, et cetera, et cetera. And you can, in principle, see the kind of lack of those details in the stepwise movement of the galvo as it's tracking the animal through this particular step. But overall, it's been, I think, relatively successful so far in our ability to mash these different techniques together. And we have I think, good outlook for getting flying insects in there after after we can get some more time in the lab. And so in principle, we hope to track the detailed limb motion, detailed body motion of animals as they're moving through structured environments, and the sort of equipment acquisition, as well as the work on this was really in thanks to KIBM and the IRG award. So thank you.
Great, thanks, Nick. That was very nice. Impressive technological accomplishments there. Let me ask a question about the acquisition speed. So 100 hertz from some points of view seems to be fast enough, but I'm wondering if as you go forward and they're actually, the animals are flying, whether you'll want to acquire at even higher speeds. And this leads to a question about the development of galvos. I know back in the early days, confocal microscopes were using galvanometers to sweep the lasers across the image. But now there are acoustic ways of going even faster. And I wondered if you and the team are thinking about going to higher speeds and maybe with different technology.
Yeah, absolutely. I think that's a great point. I think 100 hertz is sufficient for let me say, relatively directed maneuvers, as if the original video of a bee flying through, from left to right within the field of view, but certainly not for maybe the more interesting stop and lateral sideslip type behaviors. One of our planned updates to this is to have a parallel imaging stream coming through the galvo that we can use to direct tracking of the galvo, right? So people have done this before in fly work where, you can use, for instance, a quadrature photodetector that, can work at extremely high rates to direct the XY movements of the galvo so that you can keep an animal in frame. And I think those are definitely things that we want to do next, is to improve that tracking rate.
Great. Well, let's thank Nick again. Very nice story there. Good to hear that. We'll turn now to Ryan Place in Cognitive Sciences. Ryan's going to tell us about Subiculum neurons registering multiple orientations to support spatial concepts during path network navigation.
Yeah, kind of a long title there, but hold on. Okay, can you see it?
Yes.
All right, so just to jump right into things here, we like to study regions in the brain that have been traditionally found to map spatial positions in rats as they're running around freely open situations, but we want to see if we can identify features that are important to navigation, and we think navigation requires a combination of things, including having sort of a spatial perspective, being able to identify your orientation within the world that you're in, but also being able to move through the world as well and sort of reorient yourself along the way. And so just kind of a simple situation might be if I'm going up to UCSD for work anyways, and I get lucky and I find a parking spot with this green X's right here, I just have to walk over to the CogSci building, and I might kind of notice as I'm walking over the sun god statue, and at first it's kind of off to my right, and behind it is the faculty club. As I continue walking, it gets to be directly beside me, directly off to my right, and behind it might be sort of the tree forest pathway situation going on there. And then I can kind of navigate across the parking lot and get to the building that I'm supposed to work in. And so in that situation, I'm capable of moving and then re-identifying my orientation relative to a common object and knowing that I'm traveling in the direction that I was planning on going the entire time. And so that's sort of a complex process that we think involves multiple different reference frames. So traditionally, when people study spatial mapping in rats, as I was sort of saying, they'll look at regions like the hippocampus or medial-internal cortex, where a Nobel Prize was awarded a few years back. And they'll look at situations like this, where animals just freely foraging around an open arena space. Black here designates animal position tracking, and red is where the neuron is spiking. In this top figure here, we have a hippocampal place cell. The neuron is spiking in one particular location. Bottom here, medial-internal cortex grid cell. There's a periodic firing patterns that are displayed with the spiking red dots there. So, I mean, certainly there's a degree of spatial processing going on in these regions, but we think that there's more steps are needed in order to appropriately navigate a complex environmental setting. This is probably not a behavior that you would exactly employ for trying to get to work. But more work has been done in other regions of the brain that have investigated route running in particular. And this here is an example of a retrosplenial cortex neuron, but also posterior parietal cortex has similar phenomena there. In this situation, if you look at the top figure, it's a plus-shaped route that the animal is running on. You can kind of see the firing rates here, where red colors are hotter colors where the neuron is more active, up to 30 hertz. You see a periodic firing pattern there where there's 3 peaks. If you look at the linearized firing of that same neuron, you can see the 3 peaks are responding sort of to when the animal's within a left-hand turn of the maze trajectory, but also that the peak is being modulated by the placement on the route as well. So there's 2 different kinds of references at minimum being incorporated in that same neural firing pattern. And so we think things like that are useful for navigation. And so we actually want to see how these 2 systems might be influencing each other. And by studying a region that sits between them, the subiculum, it's a primary output region of hippocampus, which often gets neglected as people just study these cool looking place cells in CA1, but subiculum receives great input from these place cell regions, but also is reciprocally connected with all of these regions as well. And so that's the reason we were interested in studying. Our hypothesis would be that studying a complex situation that involves having these routes positioned in different spaces and having the animal being able to memorize which routes are used for a given location might involve something like subiculum, and the subiculum might reveal some of its processing under those situations. And so that's exactly what we want to do. Just to get into my aims for the task that we've developed, we'll go over a task that allows us to study these complex routes that an animal might memorize and be able to run ballistically, but also investigate his ability to remember these routes that are spread across different parts of the maze. And I'll get into this maze in just a second. This is what we call the hex maze. It's the task we'll be talking about today. We also want to investigate subiculum firing patterns using tetrode recordings from the subiculum and seeing how those might come to represent this situation where he has to remember these different pathways that are spread across a maze environment. And finally, kind of talk about some future directions that we would like to go. The subiculum in particular has an interesting connectivity with hippocampus and cortex that is just beginning to be known, really. And so we'd kind of like to investigate how our functional patterns might be involved in common or different circuits within that connectivity. So to get into the hex maze, the game that we're playing here is the route. The rat is going to be cued to start at one of 12 positions along the perimeter of the maze, which is laid out sort of like a clock there. And the game that he's playing is that if he's cued to start at one of the edges, one of the blue or odd numbers here, he's going to run just a small triangle like this, the smallest possible space in the hex maze. If he's cued to start at one of the green even numbered corner situations, he needs to remember that he has to run a larger triangle. And of course, this is going to repeat as you go along the perimeter of the maze, except for all of these shapes are going to be in different directions. And so he's going to have to remember that. And just to make things, just because we're nice, we're allowing the rat to run one of 2 different triangles from any site. So he has 24 different possible routes that he's trained to run. He gets very good at this. He'll do 150 or more trials a day. And sometimes with very few errors, is capable of doing this sort of navigation task. Rats basically love to run around maze spaces like this. So first, we want to investigate to what extent this type of behavior might depend upon the maze structure itself versus distal cues in the room that things like place cells and grid cells have been known to be locked to. And so what we can do is rotate the maze within the room. If we rotate it 30 degrees, actually, all of our odd numbers will now be where our even numbers previously were, and will create this conflict in the rat's head. And he maybe won't know which path to run, or maybe it's kind of an experimental question for us at the time. Maybe he just won't be able to perform the task. And it turns out that he gets very poor at performing the task at that point if you disorient the maze from the distal room cues. We allow him to use all the cues in the room, and our experimental rooms, the walls are all done up in particular ways. You might find a Jimi Hendrix portrait on one wall, and waves or picture of a surfboard or something on another wall. So he's using those cues in a particular way to solve the maze. And we can also do another sort of control if we rotate it 60 degrees, such that the maze kind of realigns with the way that he learned it in the room, just to test to see if there's something particular he's learning about the internal structure, if he's leaving odor cues or something like that in order to help him solve the maze. And he's fine under these situations. He doesn't do anything unusual. He kind of continues to track the distal room cues, basically. So now we can kind of ask, under a normal situation where a rat is performing correctly, what does the subiculum do? What do firing patterns look like there? And so here's just kind of a data bite, an example of a subiculum neuron where we have hotter yellow colors there showing when the neuron is active, and the darker colors are showing where it's less active or inactive, and just tracking locations. You kind of see, you get these elaborate profiles where there's multi-fields there. It's not clear exactly what it's representing, but it seems to be putting together some sort of structure of the maze. It doesn't seem random, basically. And if you want to look at all of the different 24 combinations and you linearize that space to see what's happening along all these different paths, you see like a peculiar structure. First of all, you see, if you look at these paths here, we have all the small triangles here on this first column, where firing rates are these little blue bumps. You can see there's basically a firing field for nearly every path, and they seem to have some kind of periodic spacing to them as you progress along these paths. They're in a different location. Interestingly, you see the same thing with the larger routes as well. The firing patterns seem to mimic them in a way. They're just, now we're dealing with larger route spaces, but you can kind of see we can see they're responding to nearly every route, and there's something about each route is slightly different, and it's unclear exactly what that is yet at this point, but we think it's probably important to him understanding which director he's on and where he needs to go. So we want to continue to investigate this to get a better picture of what's going on here. A good thing to do is to look at a full ensemble of neurons and see how that ensemble might be changing as the animal is running one of these routes in comparison to another. And so what you can do is construct one of these cross-correlation matrices. If you look at a full population of neurons, you compare sort of as the animal is running on path 2 versus itself, and see that hotter colors, which demonstrate for this higher correlation values, you get that the population of neurons is progressing along the diagonal, meaning that it's the same neurons are representing the same space fairly consistently. But then if you sort of look off the diagonal, you can see some interesting structure where the population of neurons may be also representing other features of the maze that are actually involved with different trajectories that he has to learn and run. But so you can kind of see these structures that parallel the diagonal for all these different squares here. But what's kind of interesting about the structure that you see is that it's not necessarily, often not paralleling the diagonal for any given path comparison. So if you just look, for example, at path 8 and 4 here, which would look like this shape or this potential shape, you see the high correlation value where the ensemble is relatively the same is occurring for later positions of 8, but earlier positions of 4, meaning that it's kind of like at this kind of point on the triangle, the same ensemble is doing something fairly similar. But at other points, it's different. The only time where you actually see the diagonal being striped in a path-to-path comparison of the situation is when you actually have the same temporal sequence involved in the directional components of the route. And so for this case here, 4 versus 6, you can see that the higher correlation values, the more similar the ensemble is as it progresses through the paths, is relatively the same for both of these, despite them being in different spatial locations, which is not something you would see, for example, in hippocampal place cell ensembles. So you can kind of try to test this to see which different behavioral predictors may describe this correlation matrix better, whether it's better described by say like path proportion. If neurons were firing always at the beginning or always at the end were the same neurons, regardless of what the path was, you would expect proportion to be important, and in that case, you would expect the diagonal of every square to be striped by high correlation values. Or you could maybe Euclidean space would be important, such as you would imagine just a GPS place cell type map of the environment, or the case that I'm sort of working on selling here is that maybe head directions are very important for this type of maze environment. And it turns out to be the case that the head direction predictor matrix seems to provide the most input to creating a model fit to this type of a neural ensemble matrix of the hex maze. Sort of another just more simple way to look at this is just perhaps identify all the peaks that might go along with the behavioral predictors for each of the pathwise comparisons and the peaks to this matrix. And you can kind of see in this example here, I have the peaks of the ensemble correlation matrix and these little black dots here, and the path proportion is these red lines, which of course stripes the diagonal. And you kind of just get a sense for where the proportion is not doing a good job of describing the ensemble matrix, where there's sort of offset of the red versus the black dots. You can see that there's a lot of stripes where it's just basically offset, it's wrong. And there's other points such as the 4 to 6 comparison where it's actually doing a fairly good job. Same thing with can be shown for the Euclidean space example, and you can see it's actually doing a pretty poor job. There are certain situations where you have the stars align and it does a good job. When you have everything, direction and other features line up, and then Euclidean space does a really good job, but in general it kind of veers off course. And if you look at head direction, it turns out that it actually mirrors the ensemble peaks pretty well, and you can kind of do a comparison of those 3 different features here and just show that there's less distance and so less error between the head direction to ensemble peak correlation values. Just showing that it seems like head direction is important for how the Subiculum is solving this game here. We have the animal running in this hex environment. So we want to kind of just say, like, what could be driving this? What would these neurons be potentially doing that would be driving this? And we kind of see that this neuron earlier that I showed you this 2D rate map of, it actually, which was sort of tough to tell what it was doing, it actually has very tight head direction tuning curve to it, where here we have 360 polar plot and neuron firing rates plotted in particular directions that the neuron is active, where the animal is facing. And you can see it has this, but it's not, it's clearly not only direction. There's certain parts of the maze where the animal is certainly facing that direction at points that it's not active as well. And also there, we find neurons that have multiple peaks as well. This neuron here actually does generate a pretty interesting looking 2D spatial plot where you have these kind of stripes that are striping the vertical axis in this case. And this type of neuron was actually first described by Jake Olson in 2017. You kind of see this type of neuron with 2 peaks. They're offset by 180 degrees. But we actually see lots of different neurons like this, which if you look at the 2D map, it's really kind of tough to tell what it's doing. It looks almost fairly random, although it certainly has a bunch of different activity peaks. If you look at the polar plot and the head direction peaks, you see that they have 2 really tight peaks that are facing in different directions. And so this is very interesting to us. And we kind of just looked at all the neurons that we recorded from and wanted to see like what proportion of neurons looked kind of like this. And it turns out a lot of neurons in the Subiculum during this task seem to, 25% seem to have 2 peaks that are offset in an array of different, a broad range of different ways. And there also are a bunch of 1 peak neurons. We have like 13% or so neurons that will have 1 dominant peak. We're capable of identifying these by using a model that was used by Jake Olson in his identification of axis 2 neurons. But you can kind of see over here the dispersion of the peaks, and they, as far as we can tell, we have peaks distributed throughout the environment. We have a bunch more data to update these plots with at this point, but we don't have any evidence that there's peaks are biased, and we're still looking to see, seems like a broad range of peak offsets between, there's often one dominant peak and another smaller peak. You can look at the offset between them, and they seem to have a range as well. And so that's something we're still looking into. So we kind of just want to know, like, what's the point of these? What could they be useful for? And where to go next? And so kind of the idea and the reason why we're sort of excited about it is this situation I was describing earlier, where you would need to have multiple reference frames in order to do a simple task. Just walking across campus, you might get disoriented if the statue appeared at a different place than you expected it to appear. But a signal like this is perhaps evidence that there are ensembles that are capable of switching between two different reference frames. And so that maybe the animal is capable of, often head direction neurons in an animal, just kind of aside here, have a fixed position that they fire for a given environment, but can kind of switch their position for another environment. And so maybe these two peak neurons, which display a broad range of peak offsets, can access multiple different directional maps, or even maybe spatial maps as well, and their representations. And so it's not clear if both of the reference frames would be the same level of directional, or perhaps even one of them would be more route specific or space specific. So that's something we are excited to look for as well. And of course, answering, are these neurons involved in particular circuits in the brain, is kind of our final question. And so this is where our collaboration with David Kleinfeld came in, and he performs these juxtacellular labeling of neurons in head-fixed animals. And we are in the process of developing a motorized drive based off this example here, in order to hold these glass pipettes and allow us to inject and label some of these subiculum neurons in hopes of identifying some of their projection patterns or biochemical properties, and that may distinguish the ones that we're looking at from other ones in the circuit that may have only maybe a single reference frame type of example. And so in addition to that,
are we winding up now? We're kind of way over time. So
sorry.
Yeah.
So basically that's it. Sorry about that.
Time flies when we're having fun, and it's a fascinating story. Compliments to your rats. They seem very smart to be able to do 150 trials in a day on 24 possible routes, and it'll be very interesting in the future to see how the head direction cells play out, and possibly removing those inputs will be informative. But I think we'll leave it there and turn to Charles Dickey from Neurosciences. And if you can stop sharing your screen, Ryan, and we'll bring in Charles. Charles is going to tell us about cognitive control of word retrieval in speech production through cortical layers. Take it away.
Thank you very much. Thank you, Dr. Spitzer. And thank you very much to the CAVLI for having me and for funding this work. This is work that I've been doing in the Hallgren lab in collaboration with Dr. Stephanie Reese at SDSU and Jerry Shih here at UCSD. When we speak, we're able to select from more than 50,000 words in our mental lexicon in order to produce 2 to 3 words per second. When these words compete for selection, cognitive control is engaged. So there are various models of speech production, but it's generally agreed upon that there are 4 major stages of processing. So if we see a banjo and are trying to name banjo, there's a conceptual representation in our brain. That's what the banjo is. There's the lemma, that's the word form, the phonological word form of banjo, and then there's the articulation of that word. One particular model of word retrieval is Levelt's serial processing model, which involves processing through discrete brain regions shown here. You can see the early staging processing of the lemma in yellow through phonological code retrieval and into articulatory processes. But is it the case that the processing of word retrieval is indeed serial and confined to a small number of structures? I'll note that this is on one particular side of the brain as well, the left side. An alternative model, the Dell model, proposes that in contrast to the serial model, speech is produced by a number of connected nodes represented here. And the nodes represent distinct units of speech that interact with one another, from the semantic level, which we see at the top, to lexical selection, and finally to the sound level of phonological representation. And involved in this is both lexical activation and interference, which I'm going to discuss. One question related to this, if this is true, is how focal these sequences and their connections are, and which anatomical locations in the brain are involved in processing. So the picture word interference task has been widely used to study word retrieval. In this task, participants name pictures presented one at a time that are superimposed with to-be-ignored distractor words. Here are examples of the three conditions, which is the identity condition, where the text matches the picture, the related condition, where the text is of the same semantic category, and unrelated condition, where it's of a different semantic category. Studies have shown that the response time for naming is fastest for the identity condition, showing semantic facilitation, and slowest for the related condition, here, showing semantic interference. These particular results here are from 3 patients at UCSD and OHSU, which we have a collaboration with, and I'll be showing more results, electrophysiology results from these patients later. One proposed explanation for the differential response times in the picture word interference task is that the identity word facilitates, whereas the related word inhibits target lemma selection due to lateral inhibition among coactive lemmas that are competing for selection. However, this is at odds with the finding that the amplitude of the N400, which is a strong negative potential at around 400 milliseconds, that we see represented here, is elicited in response to words, and it's elicited in response to words. It's largest for the unrelated condition, which we see in blue, compared to the related or identity conditions. So this finding suggests that there's an initial kind of semantic facilitation effect at the time of the N400 by both identity and related words, which contain useful semantic content for generating that word retrieval. So essentially, the network is being activated more easily when there's more relevant semantic content. But if this is the case, then there should be some kind of interference happening later, since the response time is slower for the related condition. So few direct invasive recordings from the human brain have been used to investigate the mechanisms of word retrieval. Since scalp recordings miss focal and deeper activities within the brain, an intracranial study is better suited to determine whether word retrieval is focal, like the Levelt model proposes, and if so, which brain regions are involved, and which are involved in facilitation and interference, which requires a short time scale to analyze. So we collected intracranial macro and microelectrode recordings from patients with intractable epilepsy. I'll be focusing on the macro recordings today. And we analyzed their bipolar local field potentials, as well as high gamma activity that indexes unit spiking, and time frequency representations. Here's an example on the right of a preoperative MRI used for surgical planning in grayscale with a superimposed CT in green, where what's highlighted is the depth electrode going through STG into the medial temporal lobe. And you can see along this electrode, there are numerous macro recording contacts. And the data I'll be reporting are from these types of contacts, the recordings done there. A single patient will have multiple of these electrodes for determining where seizures are coming from. We found widespread 400 millisecond activity following stimulus onset that was distributed throughout the brain. So this is the mean local field potential with standard error mean for our 3 conditions identified at the bottom right. And time 0 is when the image and text onset. And you can see that around 400 milliseconds, which is the time of the N400, there's a very large activation, considering this is a bipolar recording. This, by the way, is in the right anterior insula, which is indicated in the bottom. And we also found that there were high gamma modulations that tended to track these 400 millisecond activities, either increases or decreases in high gamma that are suggestive of changes in local firing. Here's another example. The previous was showing the insula. This is showing right posterior cingulate of a response at this time. Note that this is not distinguishing the conditions like I mentioned earlier. And we saw this in numerous other regions. And the high gamma here, for example, is a little more flat, so the local spiking is variable. We did also observe in certain regions that there were differential characteristic N400 responses where you see this classic response where the unrelated is largest and the identity and related are smaller, suggestive of some initial semantic facilitation occurring. And we can see that there were changes in local firing most likely occurring at this time too. And this is in the right inferior frontal sulcus. Notably, we found in some frontal sites that there's widespread 400 milliseconds activity, as I showed before, followed by a later differentiation of the conditions. So here on the right, we see from negative to positive the identity condition, the unrelated, and the related, which causes the slowest reaction time. And so this suggests that this is a time during and a location where interference may be mediated during word retrieval, and it's particularly at a later time. I'll note that the words were spoken typically 1200-ish milliseconds. And so this is right superior frontal sulcus. We also observed an interesting effect in the superior temporal sulcus. So now we're frontal, now we're going back to temporal lobe. And this was another late response where the latencies of the responses in the brain tracked the vocal responses of naming the word. And so in conclusion, there's widespread and prolonged rather than sequential serial type activity that the Loughnan model proposes. At least this is the evidence that we've found so far, interactions for facilitation and interference during word retrieval. There's an increased N400 amplitude to identity and related conditions when we record from within the brain. And this is suggesting an early semantic facilitation according to semantic content that occurs in certain regions. So if we go back to the Loughnan model on the right, we saw these broad spread 400 millisecond activations where some of them, particularly in more frontal sites, had characteristic N400 response. And we saw both early semantic and late interference effects in frontal cortex, and both early semantic and late response related in temporal cortex. So this kind of paints a more complex picture that there are a lot of different brain regions that are involved in naming that are mediating these different effects. So there's a lot more that needs to be studied to figure out what's going on here. One thing in particular that we want to do moving forward is to investigate how word retrieval is mediated within cortical layers, as well as within the layers of the hippocampus using microelectrode recordings. And we hope to have new results on that soon. We were a little slowed down due to COVID with some manufacturing restrictions and cases being postponed, but new data coming there soon. Thank you very much to the following for the support.
Thank you, Charlie. That's fascinating. So I think there are going to be a number of questions here. Let me lead off by asking whether the regions that you just identified there in your last slide, are those ones that you would have predicted or would have predicted to be involved in the identity facilitated N400, or were there surprises here? I mean, we have some knowledge, or we think we have some knowledge of the function of these different areas that might lead to hypotheses about where the N400 would be facilitated. And do your observations largely confirm our previous understanding, or do you get a dramatic new insight? You never expected this particular area to be involved.
I think it is not surprising that we see the N400 being pretty widespread, as it is a network phenomenon it's known that seems to involve multiple brain regions from various EEG and MEG studies. I think one thing that was interesting was that in some cases it's a 400 millisecond delay that distinguishes between the different conditions, and in some cases it doesn't. So it's clearly there's something going on at 400 milliseconds in different regions that are receiving those inputs, but only certain ones seem to be contributing to that initial facilitation. As far as the specific regions we identified, we did have good electrode positions here where we expected to see early effects in temporal lobe, some early sensory effects, and we expect to see the this information projected to frontal cortex, which is regulated, which probably is involved in a suppression type effect to suppress that interference, as well as anterior cingulate. But it was, I think, surprising at the same time how many areas were implicated at once and over time.
Great. Lots of questions here, Charlie. I'm not sure we'll get to all of them, but Eric wants to know whether you can say a little bit more about recording through the different cortical layers.
Sure, yeah, I'd be happy to do that. So we have, let me click through here. So this is an example schematic of a micro-macro electrode, which is a new type of electrode that we've started collecting data from. So this is a blow up of left temporal lobe, and we can see that there's a depth electrode that goes through the superior temporal gyrus into the medial temporal lobe. And the black contacts represent the macro contacts. The pinks are the laminar microelectrode arrays. And so the pink arrays with contact space at 250 microns are designed for sampling from cortical layers. And these electrodes, the microcontacts, by the way, are flush with the shaft, so it doesn't pose any significant additional risk to the patient. And these have been successfully implanted and recorded with at UCSD, OHSU, and MGH. We have started to analyze some task data from this picture-word interference task, showing possibly some later effects. So this is showing the implantation of the micro-macro electrode, and we have a laminar that appears to be in the subiculum based on the imaging, and STG. And I have superficial layers of STG plotted at the top, as well as subiculum here. This is the same task as before, like I said, so maybe there's some late activity going on, but we really need to do more work here and collect more data to say what's happening.
Great, great. Stephanie Reese wants to know, have you looked at the induced brain responses as well?
Yes, so there's data showing that there will be effects not reflected in the ERPs, because the ERPs necessitate that there's event-related phase locking. So we've looked at some spectral activities as well. So time frequency plots lock to the stimulus. And this is one example where we see for the different conditions, there is a late high gamma increase in around response time. This is in right superior temporal gyrus. And then there's some other interesting things going on where we see probably the N400 represented in lower frequencies and a beta suppression that occurs with a gamma and high gamma increase. And so we want to look at this more and try and tease apart what's going on in terms of the processing related to the different conditions, but we need to do more analysis and collect more data there.
Great, great. I regret to have to leave it there, Charlie, because there's more questions. I hope people will reach out to you on the break here, but
That'd be great.
Really a great presentation. Thanks so much. We'll turn. We're approaching noon and the conclusion of our morning symposium here. I want to thank all the speakers. Great presentations, exciting data, and so satisfying to see the collaborations throughout the La Jolla community that are really producing exciting data and identifying new directions that we think will catch fire and be very, very productive. So many thanks.
Nick, I have a few brief words, if I may. Do I have the podium here? I'd like to share a letter that I received from Nick on January 27th, 2004. How many of you can actually retrieve something that is 16 years old? Okay. Friends, it starts. With the endowment of the Kavli Brain Mind Institute in hand from the Kavli Foundation, we are pleased to inform you that Acting Chancellor Marcia Chandler has generously funded our operations during the first few years while the return on the endowment builds up to steady state. This is a terrific endorsement of our enterprise and will enable us to get off to a brisk start. It's time for us to get started, we want to convene our first meeting to identify the activities that will launch the, it was actually, it's interesting, apparently it was the KBMI, or
Yeah. Early days.
Early days, okay, it mutated in the process, and begin to define more clearly some short and long-term goals. Sign Jeff and Nick, and these two leaders really have been extraordinary. We lost Jeff several years ago, Jeff Ellman, but they worked together like, you know, like two hemispheres. It was really wonderful to see that unfold. And I think that the talks we heard today, which were extraordinarily exciting and promising and provocative, is a testament to the fact that this community really has responded to their leadership. Now, things have also changed a little bit over the years. The Salk Institute joined the KIBM, and Rusty was the, who's now the president of Salk. We took over the leadership here. And now the leadership is going to change again. And Nick alluded to this at the beginning, that we are, as of July 1st, which is coming up very soon, Yishi Jin and Ed Calloway, representing the Salk, will be the new leaders carrying forward. They have a very high bar here that they have to achieve. And this is really extraordinarily exciting time, I think, for neuroscience in general. But you know, what we heard today is that the new techniques that really are creating a real bridge between the cognitive and the cellular level, and that's going to continue. Now, I want to say just a few more things. First, Nick is not going to fade away. And in fact, he recently got a big NIH grant, an 8-year. How many of you have ever had an 8-year NIH grant? So that means that a lot more research to be done. But he's also going to be joining our cognitive neuroscience training program as a new participating faculty. So this is a new leaf in his development over time. And let me just tell you a few things about what to expect this afternoon after the break. We are going to have a symposium. This is sponsored by the National Institute of Mental Health, and Marga Behrens and Youngkook Lim will be giving symposium talks. And then the keynote talk at 2:30 p.m. will be given by the director of NIMH, Joshua Gordon. And this, I think, is really going to be a first for our annual retreat, to actually have the director of an institute telling us about challenges and opportunities in psychiatric neuroscience. And afterwards, there'll be an opportunity, 15-minute opportunity for Q&A. And so this is your opportunity to ask the director of NIMH critical questions about, you know, where are we going at NIMH? Okay, so finally, the Kavli Foundation has been extraordinarily generous over the years, and this is so important. There are 7 neuroscience centers. Each one went off in a different direction, doing different things. Our center did something really quite unusual, because almost all the funding have gone for community affairs, events. There's the Symposium series. There's all sorts of events for postdocs and so forth, but especially the very generous innovative grant program has jump-started many projects. And I think the secret is to really fund the postdocs and students, who actually are the ones who have the innovative ideas. So that's been very successful. And as part of that celebration, I'd like to introduce a mystery guest from the Kavli Foundation.
So, greetings. A mystery guest would be Chris Martin. So I'm a co-director of science for the Kavli Foundation. And if one of the hosts can turn on screen share as well, I would also wanted to dig up a thing from the past as well, because Nick has been truly central to not just, you know, what's been happening at what used to be BMI rather. I don't know if body mass index scared you away at some point.
Nice.
But now the Kavli Institute for Brain and Mind. But he was the founding director back in 2004 of the Kavli Institute for Brain and Mind. And so you can see here, this is a picture from back in 2004. This is a younger version of Nick up here in the upper right-hand corner, and Jeff Ellman, as we already mentioned. And Nick has been absolutely central through the years since, along with other leading figures at some of those very first set of neuroscience institutes. So you can see Eric Kendall and Pashka Rakik amongst this. And it really is in that set of institutes and leadership amongst all of neuroscience. Some of you may not know, but it's not just leading things at the Kavli Institute, but it's also been in the more public outreaching side. So Nick was also the founding editor-in-chief of BrainFacts. And BrainFacts is another Kavli. Gatsby is also involved with the Society of Neuroscience. And so it's really been on the scientific side, the public outreach side, and it's been absolutely wonderful working with you, Nick. And we're very much looking forward to working with the new leadership as they come on board as well. So thank you, and it's been really fun listening this morning, because the great science you're all doing is really evidence of what's wonderfully happening down in the La Jolla area. Thank you.
Many thanks, Chris.
Could I say a few words, Nick?
Of course, Jen, yes, yes.
Really is as incoming co-director for a few days from now. Wanted to thank you, Nick, for setting up this incredible high bar that we're all going to have a lot to catch. And thank you for building many programs that united the campus. Really also, you know, just many thanks and many things to learn from you. And I also want to thank Kavli to recognize the UCSD strengths and in neuroscience, and we look forward to work with you.
You're going to leap over that bar and clear it by many feet, Jen. It's going to be wonderful to see you and Ed running the show. Okay, I think that's it, my friends. Thanks so much for being with us. We have time for a quick break. Grab some lunch, get another cup of coffee, and we'll be back, as Terry said, at 12:25. Fabulous afternoon program. Terry previewed it. It's going to be terrific. So we'll see you then.
Pause. Are we pausing? Oh, actually, we're leaving.
I think we're going to leave and come back at 12:25.
I see.
You guys can all stay in the meeting if you'd like.
Thank you, Megan.